Hey guys, so my questions were, what is the end replication problem? What does the cell do to prevent linear DNA from shortening each time it replicates? And what is the importance of this? So first of all, circular DNA doesn't have an end replication problem because the DNA replication occurs in the 5' to 3' direction, and the 3' end of the leading strand grows until it joins with the 5' end of the lagging strand that comes around in the other direction. So the last bit of DNA to be synthesized, which is the replacement for the RNA primer of the last Okazaki fragment, can be added to the free 3' OH end of the leading strand coming around in the opposite direction. But that's not the case for linear DNA. So for linear DNA molecules, because DNA polymerases can only add nucleotides to the 3' OH end of a pre-existing DNA chain, there's a serious problem. So when a growing lagging strand reaches the end of the DNA molecule, and the last RNA primer is removed by a 5' to 3' exonuclease, the final gap cannot be filled because there isn't a 3' OH free at the end for the deoxynucleotides to be added to. So linear DNA molecules are therefore in danger of yielding shorter and shorter DNA molecules each time they replicate. So I'm going to explain that end replication problem to you guys in a little bit more detail and show you a figure that I drew that illustrates that. These are some figures that I drew out for you guys to show what I've been explaining. And so the figure on the left is what I'm going to talk about first, which shows the end DNA replication problem. So DNA replication is initiated at the origin, which you can see right there. And then the replication bubble grows as the two replication forks move in opposite directions. So you can see these green pieces are going to be the lagging strand, and this purple piece is going to be the leading strand. But you can see how this one is going that way and this one is going this way, both in the 3' to 5' direction. So finally, one primer, so you can see that that's the red parts. So one primer remains on each daughter DNA molecule. So you can see that here. Then the last primers are removed by a 5' to 3' exonuclease, but no DNA polymerase can fill the resulting gaps because there's no 3' OH available to which a nucleotide can be added. So you can see that the exonuclease took out the primers, and then you can see there's gaps left over. So because there's no 3' OH available for a nucleotide to be added, each round of replication generates shorter and shorter DNA molecules, which you can see down here, they're just getting smaller and smaller. So this end replication problem is solved differently by viruses and eukaryotes. So in some viruses, like bacteriophage gamma, the linear DNA forms a closed circle before it replicates to avoid this problem. In others, they use more exotic reproduction strategies, but the DNA polymerases still always progress in the 5' to 3' direction, adding nucleotides at the 3' end where there's a free OH. In eukaryotes, this problem is solved by locating highly repeated DNA sequences at the terminal ends or the telomeres of each linear chromosome. So these elements consist of short repeating sequences that are enriched in the base guanine in the 5' to 3' strand. So for example, the sequence TTAGGG is at the end of human chromosomes. Human telomeres typically contain 100 to 1500 copies of the TTAGGG sequence repeated in tandem. These non-coding sequences at the end of each chromosome ensure that the cell will not lose any important genetic information if a DNA molecule is shortened slightly during the process of DNA replication. So this is an important problem to fix because if DNA were just getting shorter and shorter, then it would get into the coding parts of the DNA, not just chipping away at the non-coding parts, and we wouldn't be here today. There would be just no possible way for the cell to continue to replicate if the pieces of DNA were so little. So the way in which these bases are added to the ends of DNA strands is through a special DNA polymerase called telomerase. So telomerase can catalyze the formation of additional copies of the telomeric repeat sequence, thereby compensating for the gradual shortening that occurs at both ends of the chromosome during DNA replication. So Elizabeth Blackburn, Carol Grader, and Jack Szostak received a Nobel Prize for the fundamental contributions to understand the telomere and telomerase. So telomerase is an unusual enzyme composed of RNA and protein, and it was first isolated in the protozoan Tetrahymena. So the RNA component contains the sequence 3' AACCC to 5', which is complementary to the 5' TTGGG to 3' repeat sequence that makes up Tetrahymena telomeres. This enzyme-bound RNA acts as a template for creating the DNA repeat sequence that is added to the telomere ends. In many eukaryotes, the 3' end of the DNA can loop back and base pair with the opposite DNA strand, generating a closed loop that protects the end of the telomere. So I'm going to show you guys this in the other figure that I drew. Okay, so I'm going to talk about the figure on the right, which shows the action of telomerase. And so first I drew eukaryotic DNA, and then down here it shows the end of DNA, which is called a telomere. And so if you imagine this zoomed in right here, we start with the telomerase binding to DNA, and then here I show the two components of telomerase, which is the RNA component and then the protein component. So nucleotides are added to the 3' end of DNA. So as you can see, this was 3', this was 5', so this is 3' and this is 5'. So nucleotides are being added here. And then the telomerase advances along the DNA strand as more nucleotides are being added. So you can see that down here. It advances that way as these nucleotides keep getting added to their corresponding base. And then standard replication machinery extend the complementary DNA strand. So you can see that here. And then in many eukaryotes, the telomere DNA loops back on itself to cap the end of the chromosome. So you can see that here. So in multicellular organisms, telomerase resides mainly in the germ cells that give rise to sperm and eggs, and in few other types of actively proliferating cells. The presence of telomerase allows these cells to divide indefinitely without the telomere shortening. In most cells, telomerase is inactive. So this places an upper bound on the number of times such cells can divide, because at each successive division, telomeres become shorter and shorter. So for primary cultured cells, which are typically used in scientific experiments and for lab work and studying diseases, this limits the number of times cells can be passaged, so meaning over how many generations the cells can be propagated. This limit, which is 50 to 60 generations for many cells, is called the Hayflick limit, named after Leonard Hayflick, who investigated this growth limitation in the early 1960s. So if a cell divides enough times, the telomeres and the descendants of the cell are in danger of disappearing entirely. And then the cell would be at risk of eroding its coding DNA, just as I said earlier. This potential danger is averted by an orchestrated cell destruction pathway set in motion by shortened telomeric DNA, known as apoptosis. So immortalized cell lines like HeLa cells, which we studied a few chapters ago, that were isolated in the 1950s, circumvent the Hayflick limit because they produce telomerase. therefore allowing such cells to be passaged indefinitely. And that's why HeLa cells have been so crucial for a lot of research, specifically with cancer. So cell death triggered by a lifetime of telomere shortening is thought to contribute to some of the degenerative diseases associated with human aging. For example, increased susceptibility to infections due to the death of immune cells, inefficient wound healing caused by depletion of connective tissue cells, and ulcers triggered by the loss of cells in the digestive tract. So people who inherit mutations to telomerase typically experience similar degenerative changes and show symptoms of premature aging, and then usually die at a pretty young age because of the telomere shortening. And because of this relationship, a lot of scientists are thinking that telomerase-based therapies could be the key to combating human aging and possibly extending lifespans, which that's a pretty big discovery. And I'm curious to see if there's really extensive research being done for this, because once people hit like 30 or 40, they're looking for skin treatments with retinol and all that to try to get the collagen stimulated in their skin. And if things like ulcers and other degenerative changes could be helped by telomerase therapies, I think that would be pretty revolutionary. And with healthcare, we've already extended the lifespan by way more than it was even 100 years ago. So I'm wondering how far we could take it with these telomerase therapies. But thank you guys for watching. Let me know if you guys have any questions, and I'll try to answer them to the best of my ability.